Hello, my name is Manuel Niell. I am from Andorra Research and Innovation, and we are going to talk about the two endangered species, Helleia and Coenonympha glycerion, that are two endangered species in Andorra and other places. First of all, we want to introduce you where is Andorra. Andorra is a small country in the middle of the Pyrenees, between Spain and France, as you can see in the picture in the left and in the right. Our territory is about 468 square kilometers, and 58% of the territory is above 2,000 meters of altitude. It is an altitude from 1,000 meters to 3,000 meters, almost 3,000 meters. So it means that we have in a small territory a great amount of habitats and different kind of microclimates. Then we have a big biodiversity. There is also a big problem with the climate change because these mountains are in the middle of the Pyrenees, and it's expecting that the mountains and the Mediterranean areas are expecting to have more impact the climate change. Then our center have different bioindicators, like for example, the butterfly. We have 10 transects, and we are following the methodology of the butterfly monitoring scheme. About the climatic change, in the left, you can see this graphic about how the temperature increased in the last decade, about 0.14 degrees, and also the annual precipitation has decreased about 4%. About the transects that we are following, one of the most important, it's here, one that's called Bordas d'Ambalira, when we found two species very interesting for the first time in Andorra last year. For example, it was, Helleia helle, also known as Lycaena helle, and also Coenonympha glycerion. The Lycaena helle is the third population known in the Iberian Peninsula. Let's talk about Helleia helle. Helleia helle belongs to the family Lycaenidae, and the European status of UCN are endangered. It's a boreal-montane butterfly adapted to moist climate conditions. It's a rare butterfly, and it's very interesting because their populations are strongly isolated and the dispersal ability is limited. Also, we can find in different these moist meadows, peat bogs, etc., but the caterpillars fits with Polygonum bistorta. It's interesting that the populations had a fast decline before 1995, although it seems that now the populations are more stable. Here you can see the distribution of these species. You can see that the north of the Iberian Peninsula is the meridional limit, occidental meridional limit of this species. Other species is Coenonympha glycerion. It's from the family Nymphalidae, subfamily Satyrinae. The European status of the UCN is the less concern, although in Spain recently, or in the Iberian Peninsula, sorry, it suffered a strong regression recently. So in Andorra also, it's identified as a threatened species as in Spain at this moment. For example, in this map that we are showing you now here, is not represented Helleia helle because, as I know, it's unknown in Catalonia, and there are only 3 populations in the Iberian Peninsula. Here it's only Coenonympha glycerion. You can see that Coenonympha glycerion is these black dots in the picture of the left. And in the picture of the right, you can see that the dynamics of these populations. In red is the species that it's decreasing, and in blue, the populations that are stable. Both species probably are in danger because are threatened because deforestation, eutrophication, water extractions, drainage, overbuilding. We still are not clear why these species are threatened. Well, we decided to use these 2 species because they are threatened, as I told you. Both species were collected at the end of June to ensure that they finish their reproductive life. The species were collected in the field, directly alive, put it in the fridge, frozen at -80 degrees. The genomes of this species has been assembled by the SINAC up to EBP standards. Long red seed DNA sequencing of wing and leg tissues using Oxford Nanopore was done at the CRG. Using an innovative method for long read transcriptome analysis, the CRG is currently producing a highly relevant annotation that can be used as a reference within Lepidoptera. Here we have the results, first of all, about Coenonympha glycerion. Coenonympha glycerion, it's a highly heterozygous, has a highly heterozygous genome, 2.7%, 106 gigas of high quality on reds with 38K read N15 were produced. The data were assembled with both fly and next de novo, with the latter producing the most continuous assembly. The assembly was polished with HIFO using long and short red data and purged with purge DAPs. OmniC library sequenced the 459 megas. Assembly was scaffold with JHEs into 29 chromosomes. Counting N15 is 12 megas, scaffold N15, 16.5 megas. And the quality values are 45.6. The assessing genome assembly and annotation quality was made by Busco with these results. Here you can see on the right the graphic with all the reads, the genoscope profile, and the high C diagram. Here also the same with Helleia helle. You can see that it's less heterozygous with 0.5%, 100 gigas of high quality on reds with 42 keys. Red N15 were produced. The data were assembled with both fly and next de novo, with the latter producing the most continuous assembling, well, more or less similar to other species, resulting in a 547 megas assembling 25 gap-free chromosomes. Cotic N15, scaffold N15, 20 are the same in 23.6 megas, and the quality value 46.6. Well, about the RN sequence annotation, sec annotation, this slide contains the RN sec annotation, RN sec data results for both species. On the left, the workflow for sample preparation, sequencing with on and data analysis, using the nanoplot in green, in yellow lyric, and Genate in orange. On the right side of the slide, the tables with the tables with the statistics on sequencing and annotation steps. In green, the nanoplot quality control data of nanopore reds showing a good median quality score ranking from Q11.7 to 13.1, depending on the sample and tissue. N15 red length is around 1.5 Ks for all the samples, with the exception of Helleia helle wing that is a bit lower. To generate transcript models, data in yellow, CRG lyric pipeline was used and Genate was used to generate gen predictions. This method is described in another talk in the computational method section. In purple, the overlap between these two methods. Even though the analysis for Coenonympha glycerium is still in process for the Helleia helle results, it is clear that we are able to identify genes and their potential functions using different approach. This will allow us in the future, this will allow future comparative and population genomic studies. And that's all, thank you.